So let's start.
Hello.
Hello.
Yeah, Joa. Shall we start?
Let's start, sir. Yeah, sir. Let's start.
Or we'll wait for few more minutes? No?
Yeah, sir. We are audible.
Yeah, okay. Hello everyone. Good evening. I am Dr. Sharat Chandra from Great Eastern Medical School and Hospital. Yeah. In this second session, we will discuss about this, how we will do this methodology part of the research. Designing methodology and working on it and publishing our paper. In our previous session, we have discussed about fundamentals of research, why it is important, how to formulate a research question, and everything, right? Now, we will discuss about the methodology, writing the paper, and publishing your work. Okay. Can you go to the next slide, please? In this session, what we will see is the research methodology, how we will do data analysis and statistics, interpreting these results, writing a research paper, and final presentation. Okay. Okay. Now, what is research methodology? It is like a blueprint for your project. It is like defining your research question, ensuring reliability. It will help in reproducing our findings in a good way. It will provide a clear roadmap. It will give you. It will. It's like it's a. It will pave a path. It will pave the path. This methodology, it is of this study design. It is of two categories mainly. It is like observational studies and interventional studies. In these observational studies, yeah, can you go to the next slide, please? Types of, yeah, these quantitative research, yeah, before those observational intervention, yeah. Now let's see about types of research methodologies. These quantitative, qualitative, mixed methods. In that quantitative is we will discuss. It's all about math. The descriptive, including math, is qualitative. Sorry, quantitative, and only the description, only the narration part is qualitative part. And mixed methodologies, it's like we will include both the description and also the mathematical part of it. Now we have to see which type of methodology will fit into our work. Okay, go to the next slide, please. In this, I don't know how many of you can see it clearly, but what it is in this is first, the dark one is clinical research. It is of two types, either quantitative or qualitative. In that quantitative may, which descriptive and analytical. Can you zoom it once? If I can, if I think maybe the members can see. Joa, can you try to zoom it? Is that possible? It's not clear. No, the previous slide. Yeah, I think I'm sorry for this quality, guys. It's a screenshot and it's not clear. It is in the descriptive may. See, move it to the left side, please. Left part. Yeah. The case reports, case series, and in analytical part, there are two types, either observational and experimental. In observational studies, what we will do is these cohort studies, cross-sectional studies, all these retrospective, prospective studies, everything. It is like we will see, we will observe everything. The type of observation studies, these are cross-sectional, cohort, and case control studies. We will observe what's happening, we will observe what happened, and we will write. We will write the report. Experimental studies are the randomized control trials and clinical trials and quasi experiments. It's like we will work on them, we will test everything, and we will prepare the report. Go to the next slide, please. Yeah. The main components of research methodology, we have to select it's experimental or observational. We have to concentrate on sample selection. Whether it is experimental or observational study, we have to concentrate about sample selection. Whether it is a cross-section study, we have to select a sample at one point of time. We will collect the samples. We will collect the data at one point of time. If it a case control study, we will follow the sample population and the whole time of our study throughout the study. In that way, we will see which type of study and we will collect the data either in interviews or surveys or lab tests. All these we will consider the variables that are affecting our study that might affect maybe independent variables or dependent variables or maybe even some confounding factors. Then we have to see whether this is ethically possible or not. Like if we do some research on pregnant women or on what is it, young below 18 years or even in children, infants, then it's we have to see ethical considerations. We have to get consent and we have to do our part. It's like well-structured methodology will enhance our research credibility. Now, in this methodology, the main thing is selecting which type of study and selecting sample. How to choose a sample? We have to define the target population. Yeah, we will consider, okay, if I am doing some work, I am doing how many students know about maybe how many students know about aesthetic medicine or geriatric medicine. No, now what I have to do is geriatric medicine is something related to medical field. Now I have to select the students from medical field. I have to define my study goals first. I should understand what are my study goals. Then I have to select my target population. Okay, now geriatric medicine. It's for, let's consider medical students. And then use probability sampling for, it's like generalizability. We have to see, okay, now I have selected medical students. Can I generalize my findings from these selected students to the whole population or not? To the whole medical students. Okay, you might have considered one college students or two college or maybe 5 college or 10 college. But can you generalize this to all medical students data? Whether it is non-profitable, or exploratory data or qualitative research, whether okay, can we do this or not. Next, sample size. We have to see there are around every year around 80,000 students are getting admissions into the college. Now I'm considering only 800 people. Can this sample size is considered as a good sample size or not? Can I consider it, or I should increase number of people, or what? You have to think about considering the sample size. What are the effects? What are the confounding factors I will discuss? What are the factors that may affect this? Or we have to see everything. It's like we have to consider all the factors that may affect the sample size. All the sample size, it depends on the prevalence. It is like if you know about one condition or any disease, then okay, you can calculate the prevalence of that, and you can calculate the sample size. But if you are doing something novel, that okay, there is no one till date, there is no available literature till date. We are the first one who is working on it. So you will not get any prevalence or any previous studies. Then you have to think, okay, now these many people are there, number of students. I have to see, can I generalize this to all the population? Am I considering all the areas? Am I selecting only? I'm confining to only one particular college, or is this a multi-centric study? I'm considering all over India, students from all over the India. Am I sending this to Google Form to everyone? You have to consider everything, and you have to think about the error level. What is the percentage of error that might occur in this? Okay, maybe 5% or 10% of people may give some mistake that is like it's not a mistake, it's not something that they want, they give intentionally. Maybe it's like it's something that's not even in their hands. It's like that is not in our hands, and we have to think about that while calculate the sampling size. Large sample size improves reliability. If you try to include more people, then everyone will think that your study is reliable, because you can generalize it to the whole population. Okay, next. Okay, sampling methods. Okay, now I want to calculate sample. Okay, I have decided my study group, medical students. Now I have to calculate the sample size. How many students? Should I consider all 80,000 students from the whole country of a single batch, or 8 members, or what? 800, 8,000, whatever it is. These sampling methods, there are probable samplings and non-probable samplings. In probable sampling, it is either random sampling. Okay, now you will just think a number. It is like okay, I am considering 500 students. Or stratified sampling, or cluster sampling. Okay, I am considering this cluster of students, and I am doing this. Or systematic sampling, I am considering, okay, these are the students I'll pick, maybe from 1 or 2. I'll consider every nth person, every first person. I'll create a group of 10 students each, and I'll pick third student, number 3 from every group. Or I'll create a group of 5 students and 100 groups, and I'll pick 1 and 3 from every group. You have to fix, and that's one thing. And convenience sampling is okay. It is like how many students are available with you. Based on your chances, your convenience, you will collect the sample number of participants. And it's similarly the purpose of sampling also. Snowball sampling is we will give this, it's like at first we will not fix about the number of participants. It is like we will give it, we will distribute the data, we will distribute the questionnaire or survey or Google form, whatever it is, to the participants, and we will tell them, please share it with your peers, share it with similar kind of people. Okay, now geriatric medicine, every first year, every fresher will not know about what geriatric medicine is. So what we will tell is, okay, the final year students, now please share this to your parallel batch students or interns to, and tell them to fill this form. Okay, now you are concentrating on the participants, the population who are similar to your criteria. You will give them to the person who fulfills your criteria, and you will ask them to share the same with the person who fulfill the same your criteria. It's like you will give it to the person and you will tell them to share to the similar person. Okay, next slide please. Okay, now you have collected, now you have chosen this sampling method, and you will collect the data either by survey or by Google form or maybe by interview, anything, and now you will get data. Why data analysis is important? This data analysis, it converts the raw data into scientific information. It will identify the trends, the significance of each and everything. It will it will explain, it will compare the findings, it will acknowledge your study. It's like it will acknowledge the findings. Okay, these people know about this, these group of people know about this, or these college people know about this. In that way, it will provide the scientific evidence. It is, data is the foundation for your research. You have to analyze it wisely and you have to present it. Next slide please. How to analyze the data? There are so many types of analytical tools, the statistical texts we can use to analyze our data. Okay, if the type of data is yes or no categorical questions, we will use chi-square tests, yes or no, male or female. Then yeah, we will use chi-square tests. If it is continuous data, like it's monitoring BP or weight or age of a person, then we will use t-test or ANOVA. All these chi-square, t-test, ANOVA, all these are different types of tests, types of statistical texts we will follow to analyze our data. And if we work on variables, relationship between variables, correlations, then we will consider Pearson test. If we think about predictive models, if we work on predictive models, we will go for regression analysis. In that way, we will use, depends on the type of data, we will select the tool and we will analyze our data. Next slide please. Okay, types of statistical tests. All these I have mentioned earlier, as I have mentioned earlier, we have seen those tests, right? For descriptive analysis, mainly we will use mean, median, mode, and we will calculate range. We will see for variations, we will see for frequencies, we will draw graphs about our data, and we will draw charts.
And to see inferential analysis, we will calculate the confidence interval. To hypothesize, to test the hypothesis, we will work on t-test, z-test, and t-tests, and chi-square tests. For bivariate analysis is two types. It is like if there are only two types of variants available, and if we want to work on them, we will go for mostly we will go for chi-square test, and we will also do the same with the t-test or correlation test. If we have a multivariate, if we want to do multivariate analysis, mostly we will go for ANOVA. And if we want to go for non-parametric analysis, it is like they are not of, generally we will not go for them. But if we have to go, then we will use a Friedman test. These are the different types of statistical tests we will use for different types of data. Common errors. What are the errors that we will do? We will face the problems we will face while doing this data analysis. Choosing different, it is like choosing wrong statistical test for a wrong sampling type, statistical type. The data you need, you have data of something, and you have chosen some other statistical test. You need answer for yes or no, and you have chosen some ANOVA test, then it won't be it won't be compatible. Ignoring sample size calculation. If it's a novel study or if it have a very less prevalence or something, some factor affecting your sample size, then it's okay. But if you have, if the condition is prevalent and there is some prevalence in the population, and still if you ignore calculating the sample size, then it will affect your data analysis. And the most important thing is data manipulation and fabrication. It's like okay, now I have considered I'm considering for 1,000 students, 1,000 medical students, and I'll collect the data. But while collecting data, you got only around 300 to 400 samples answers, and other 600 those participants are not responding. Or okay, you got 600 samples, you got 600 answers, and 300 or other 400 are not responding. Now, what they will do is they will fabricate, they will manipulate this, they will multiply, they will apply the same data to everyone. It's like they will replicate the data. That will cause an error in your data analysis. And another thing is now selective reporting and of significant results. Okay, now I am doing on geriatric medicine. What I know is, okay, as a first year student, many first year students will not know about geriatric, what geriatric medicine is. Many final year students will know about the geriatric medicine. That's my intention. That is back in the back of my brain. That's in the back of my brain. So what I'll think while doing the study, and what I think while analyzing the data is, okay, first years don't have any idea, final years do have an idea. Then I'll select the results, I'll select the responses which support my hypothesis, and I'll present these data. It's also a mistake. It will also causes data analysis mistakes in data analysis. So to prevent all this, what you have to do is choose your methodology, correct type of methodology, correct type of statistical test, and go to a statistician and use proper analytical tools to analyze data. so that you will prevent the data analysis you will prevent errors in data analysis. Next slide, please. How to interpret or okay, now we have analyzed the data. We have chosen we have decided how to do the study. We have collected the sample. We have analyzed it. Now, how to read the analysis? Generally, what we will do is we will calculate p-value. If the p-value is significant, that is like less than 0.05, then we will consider it as a statistically significant value, and we will see the confidence intervals. We will calculate the effect size, how much meaningful the lecture that the result is. Okay, I've seen this. I'm thinking that my hypothesis, I do have some hypothesis in the back of my mind. Okay, I'm thinking about this. What about the results? Are there any similarities, or as my hypothesis is contraindicating the results, or what it is? And we have to see it's like statistical significance is never similar to clinical significance. Maybe it is statistically it is significant. Maybe 100 members, 100 maybe out of 1,000 students, 900 students might know about geriatric medicine. But generally, maybe they just know about the word. It is like, yeah, they heard about it. If you ask me whether I know about geriatric medicine or not, yeah, I heard about it and I'll choose yes. But originally, I do not know what that is. I think you got it. Yeah, I heard about it, but I don't know in detail about that. It is like, it is statistically, yeah, I know. But clinically, like originally, genuinely, I do not know about that. Okay, next slide, please. Okay, now how to avoid these misinterpretations? We have to see these correlations. They are not similar to, they are not causations. We should not overgeneralize the small samples by just considering 8 or 80 or 800. We should not generalize this to all 80,000 people. We have to see, we should not include any bias or any, we have to concentrate about confounding variables, all the affecting factors while doing the study, and we have to maintain that integrity. And we have to validate the results with existing literature. We have to validate. Okay, now you got these results. Let me check what the other previous literature told about this. And we have to validate your study. Then it will be of a good use. Next one, please. Okay. Now, till now, I've got my research question. I've thought about which journal, what's my target journal. I've framed my methodology. I've collected data. I've analyzed it with correct statistical tools. I've interpreted the data. Now I have to write a paper. First, we have to give the title to the. Then normally the structure of the paper, the research paper is, we have to give title, abstract, introduction, methods, that is methodology, the results part, discussion part, conclusion, and references. but it is like this is the structure of your research paper. What you have to start first, because as title is already fixed with your IEC, your Institution Ethical Committee certificate, you cannot change your title. So you are stuck with the title, so you can fix your title. And now we have to see your methodology. Then you have to write your what I personally follow is I'll fix my title, then I'll see the research question, research gap, everything. Why I am doing this? Why am I doing this? What's the rationale? First, I have to discuss about the problem. I have to discuss about the rationale, and then I have to justify why I am doing this and why that is this research is important at this point of time. All this we have to include this in introduction. In methodology, we have to write what's the type of study, the study design, the sample size, the study setting. All these are inclusion criteria, exclusion criteria. Inclusion criteria, exclusion criteria are something. It's like okay, I'm considering all these people with these specific characters. Okay, I'm considering medical students who attended general medicine classes, or medical students who attended clinical rotations. Exclusion criteria, maybe first year students or second year students. It's like that. We have to maintain those. We have to form those inclusion and exclusion criteria, and we have to write them. Then we have to write our results from our data analysis. We have to write our results in a tables or figures or whatever it is, in pie chart, bar graph, anything. Then we have to write the discussion part. Discussion part is comparing your results with your reference articles, maybe some other articles, the results from other articles. Okay, in my study, around 60% of final year medical students know about the geriatric medicine. But someone told that in this particular study, only 30% students know about this. Maybe this is the factor. Maybe we have maybe the patient load or something, whatever. Maybe this might be the cause for this variation. We have to write conclusion. It is like summarizing your whole findings. One thing you have to remember while writing the conclusion part is you should not mention anything that is citable. It's like, okay, now in my study, I found 50% of students who know about geriatric medicine. 30% of students just heard about geriatric medicine but do not know what it is. And 20% of students never heard about geriatric medicine. Now, I can mention these values in results part, in discussion part. But in my conclusion, I should never mention these values. It's like it should not be citable. I should not pick something from your conclusion to add in my introduction part or somewhere else. We can just maintain. You have to give it in a descriptive way. In our study, we found many students, majority of the students know about geriatric medicine, and very less number of students do not even heard about geriatric medicine. You can just write it in a descriptive way, but never should be in a citable form. References. They are the literature, the similar literature according to your study. they are the similar references. We have to cite them according to the journal guidelines. Every journal have specific guidelines, specific other guidelines, specific reference citation guidelines. Maybe as we have discussed, APA style, Vancouver style, Chicago style, anything. There are different types. We have to check in our other guidelines and we have to cite them according to that. Well structured paper is easy to read and it is easy to publish. If you write it well, yeah, it is easy to read. Okay, I can read, not me, anyone can read it, anyone can read your, okay, and anyone can read why you did this, anyone can read what you did, which type of data you have collected, and what about your results and everything. Okay, and common errors in writing. Can you go to next slide, please? Common errors in writing. Too much jargon, poor logical flow, lack of clarity in results and discussion. You have to maintain the clarity. You should not confuse the reader with your results and discussion part. You have to maintain the flow. Okay, from this, I did this from step 1 to step 10. You have to maintain that flow. Okay, what you did and what you got, and you have to showcase the same thing. And you should never overstate your findings. You have to write it concisely and clearly. You have to follow journal guidelines and you have to proofread it multiple times to prevent these mistakes. Okay, next slide, please. Okay, now we have completed your paper. You have drafted it, you have prepared a manuscript. Now, publishing. Publishing is an effective way to showcase your research, and presenting is an effective way to showcase your research, because if you present it in a conference or if you present it in front of your professor or wherever it is, if you present it in front of a group of people, then they will ask you questions. They will ask you, okay, why you did this, why you have chosen this methodology, why did you included these people, why you have excluded these people, so that in that study maybe you cannot do anything, but from the next study you will understand, okay, I have to do this, I have to do that. Okay, as you will show all your work to your guide, to your statistician, to your community medicine professional or whoever it is who guides you, they will ask the same questions while you are writing your paper and they will guide you in everything. But still, if you go and present it in a conference or in a seminar or symposium anywhere, then the participants who do not know about you, the professionals, the scholars in that particular field will ask you questions, and it will help you in preparing a better paper next time. Next question, please. Okay, to how to prepare your presentation. Most of the conference will have this structure for their presentation. It is like, first we have to mention our it is title slide, then introduction, then methodologies, the result, and conclusion. It will be the structure. It will be the structure in many conferences. If we submit an abstract, the abstract should be in this format. And you have to be prepared for questions. Okay. Any questions?
If you have any questions, please raise your hand. We will call you out the names and you can ask your questions one by one.
So for everybody who has questions, please raise your hand and I'll be calling out your name. And then you can ask your question to sir. Anybody who has doubts?
You can ask any question. Don't think maybe it's just a silly question or something. You can ask anything.
So Nirmaya has a doubt. Yes, Nirmaya.
Yeah, Nemai. Go ahead. Hello.
Hello. Yeah, sir. Can you hear me?
Yeah, Nemai. Go ahead.
Yeah.
I can hear.
So, sir, I'm right now in my second year of MBBS,
Okay.
and now I have chosen a topic so that I've started working on the research project.
Okay.
So I'm actually concerned about publishing the research project. I mean, from now only because I don't think what I'll be able to do with my research project at the end. Like I don't know how to explain. What I'm trying to tell is it necessary that only some new statement has been made, or just I'm just using what is already existing, and then I have
No, yeah. Are you talking about systematic or native review or anything? Or you worked on original research or any review article?
uh, I'm working like I'm doing a prospective observational study.
Okay.
Uh,
Okay. In that, you can, okay, you found something that is similar to your previous studies. Maybe they might have found the same thing maybe 50 years back. Now it is 2025. Maybe they might have found the same thing in 1975. And you found the same thing even in 2025. Then you can emphasize that, okay, the results are similar even after 50 years. You can emphasize your results. So you can publish your work. You can say that, yeah, you did the work in that time period. I did the work in this time period. The results are same. There is no considerable changes in whatever work you did. There is no considerable changes in this. You can say, you can emphasize on that.
okay.
Yeah.
In short, small గా అడగాలంటే my doubt will be like
Yeah, Telugulo you can ask Nirmay. I can.
yeah,
Telugulo అడుగు.
అదే అంటే ఇప్పుడు ఏమైనా కొత్తగా కనిపెట్టి అది చాలా పెద్దదైతే మాత్రం పబ్లిష్ చేయొచ్చా? లేకుంటే ఏం చేసినా కూడా
No.
పబ్లిష్ చేయొచ్చా అని?
Yeah, let me translate it for everyone.
ఓకే.
What he's asking is, should I present something that is new, or something can I present the results which were already found by someone else? Yeah, you can present whatever the data you got, because others will know. It is like now you have worked on it, you are working on it, maybe you are working on it from the past 2 or 3 months, and maybe you will take 2 or 3 more months, maybe it's around 6 to 8 months to complete your work. You got something similar result as your previous previous researchers.
Or I can
Now,
get a different one also, right?
yeah, you might get different one, you
Yeah.
might get similar result. If you get if you get different result from the same population, then you can publish it saying that you and me, we both followed same methodology, same protocol, and we got different results. Maybe there might be some other factors, maybe some confounding factors or something.
Yeah.
You can say about that, or you can point out some questions. You got this and I got this. Why, Ani?
Otherwise, if you get the same results also, you can say, yeah, there is no change in similar in this point of in this group of people, there is no change. You can say anything, so that next one won't do the same thing. It will save the time for them.
Yeah.
Yeah.
Okay, Anand.
Okay. Okay. Nandini.
Then, sir, a person has asked how to select the best citation for publication.
Citation is the format how you are. It is like yeah. Just a minute. Yeah. Citation is like you are mentioning some other article. You are mentioning your reference, one of the other previous articles who worked on the same topic as you. You are working on something, and you have found some other article who already worked on same thing. Now you are mentioning their article as your reference. To cite this article, you have to follow some structure. There are different citation styles, different structures to cite an article. All these you can find that in others guidelines. In journal homepage, you will find if you try to submit an article, you will see other guidelines. In that, they will mention about the citation style, in which type they need the citation. You can follow that guidelines and you can cite the article.
Okay, I hope that clears their doubt. Dheeraja also has a doubt. You can
Hello,
speak.
sir.
Yeah, Dheeraja.
Sir, what is rationale you mentioned in presentation? May I
Yeah.
know?
What is?
Rationale.
Yeah, you have to be rational about your
Yes.
question. Are you talking about the introduction what I have discussed?
Yes, sir.
Introduction part of your
Yes, sir.
paper.
Yes, sir.
Yeah, it is like you have to say why are you doing this?
Okay.
Yeah, it is like okay, now I'm working on as considering my example of geriatric medicine. Now why I want to work on this geriatric medicine is now why I want to study this particular topic is according to recent studies from in the next 25 to 30 years, population of 50 more than 50% of India population will be in geriatric age group.
Okay.
So already the geriatric people are facing these situations in India. It is like in introduction you will mention about there is something like a funneling technique, funnel from broad to narrow. Funnel, funnel, right?
Yes.
The base and the tip. First you will cover the broader area and you will pinpoint it to something. Okay, in the whole world, this is the condition. This is the number of around maybe 50 billion people are 50 million people are in geriatric age group. Among these many people are facing these issues in different countries. You can cite covering the whole broader area, and we can narrow it down to India. In India, there are 1 crore people who are in geriatric age group. They are getting these services. They are facing these problems. You can come to, if you want to do it in your own place, you can funnel it down to your own state, maybe from Karnataka or Andhra Pradesh or Tamil Nadu, wherever you are from. Or if you want to share the Google Form to every student, then okay, now as these many people are increasing, as the numbers are increasing, I want to do this so that I want to know how many people are interested in this, or how many people want to know about this, how many people want to pursue the career in geriatric medicine, so that I want to calculate the care. I want to hypothesize something. Yeah, okay. If these many people want to, maybe around in 1000 sample, in my 1000 sample, maybe around 500 final year medical students want to pursue their career in geriatric medicine. Okay, maybe our future geriatric people might get good care, good quality care. Is that Allah? You can. It is like,
Okay.
yeah.
That is emphasizing the importance of my study. That
Yeah.
okay, sir. Thank you, sir.
Yeah. Thank you.
So Gayatri has a doubt. Is systematic review and meta-analysis different from this research methodology? Is this webinar for publishing original research paper?
Systematic review and meta-analysis different from this research methodology. Yeah, systematic review, meta-analysis. Yeah, they are different. It is like original research, it is different. Systematic review and meta-analysis, they are different. Original research is you are working on something and you will discuss about the results. The systematic review and meta-analysis is you will review the available literature. Maybe okay, now there is a drug. Some okay, take paracetamol. I want to try paracetamol. I want to give paracetamol to patients suffering with cold, and I want to see whether it is working or not. So if I do some work on it, if I start some work on it, and if I publish the results of my study, it is a kind of original research or clinical trial, you can say, or original research. Or if I do something like, okay, there are some previous studies, some other researchers who did the same thing. I will collect all the data, all the literature, all the results that they have published. I'll summarize their findings. That is systematic review. And if you summarize their findings with numericals, it is meta-analysis. So yeah, original research and systematic review and meta-analysis, they are different. Is this webinar for publishing original research paper? No, it's just like we have just discussed about methodology. It's like a broader overview of methodology. It's a broad overview. It's nothing. We haven't discussed anything specific to original research or anything specific to systematic review or meta-analysis. It is generalized.
Okay, so the next question is how I believe Vijay wants to ask how to select a journal to publish.
Is that Vijay?
He has mentioned journal selection.
Yeah, I've seen that. Vijay, are you there? Vijay?
Yes.
Yeah.
Hello.
Yeah, Vijay.
Yeah, please tell me, sir.
Yeah, what's that journal selection? Can you elaborate that?
Yeah. How to select journals? Points which to be considered.
Yeah, how to select journal. It is like in our previous session, in our session 1, we have discussed about that. The thing is, okay, now you want to work on something. Okay, as I mentioned, this geriatric medicine. Okay, I'm considering to work. I've considered. Okay, I formulated a question, something related to geriatric medicine. Now, how to select a journal, how to publish my work, where to publish my work is first I have to see which journals are accepting geriatric medicine, which are specific. Every subject, every category, every field have some specific journals related to that. If you say cardiology, you will say American cardiology, European cardiology associations. All these associations, they have specific journals.
you will find some specific journals even for geriatric medicine or whatever it is, whatever type of work, whatever the field you may choose, you will find some specific journals. You can try to submit your articles in that specific journal which is related to your field of work. You can try to publish there, or even many journals will have a scope of publication. They'll have a broad, wide scope of publication. You can even publish in that. Or if you don't know how to find all these things, then simply what you can do is if you find, okay, in your references, you might have selected maybe 10 or 20 or 30 references for your study. Then you will see how many articles are from single journal. How many of my references are from single journal? From 30, maybe there are some around 10 journals are from specific, 10 references are from specific journal. Then you can simply go to that journal and you can try to publish your work, because you found something similar to your work in that specific journal. That means that journal is publishing something similar to your work. So you can try with that. First,
Yes.
yeah, okay.
Thank you so much. Thank you.
So Nirmay has a doubt regarding data collection. Nirmay, you can
Yeah.
ask.
Yeah, sir. So my other doubt is, so now I'm started to collect the data, right?
Okay.
So what I'm willing to ask you is, after I collect the entire data, is it mandatory for having a biostatistician so that he can assess, or is it something like we can do by it by ourselves, like putting up?
Yeah, you can do it. You can do it. You can do all the statistic part on your own if you know how to deal with the ANOVA, t-test, mean, median. If you know how to calculate those. If you have a SPSS software, if you know how to calculate all those things, yeah, you can deal with your data. There is no need of a statistician, but ideally you need a statistician, because I don't know if you know, as a secondary student, I don't know if you know all about the statistics.
No, not really.
Yeah. So what I suggest is go to your statistician, sit with them, ask him to tell you, sir, please tell me how to do. I want to learn about this. I'm interested in research. So I want to learn how to do these ANOVA tests, how to calculate the results using this, how to calculate the p-value, how to calculate mean, median, everything. So, sir, please teach me how to do. You can ask him, you can sit with him, with your statistician, and you can learn from him. And you will find some certification courses also related to these statistical works. You can do the course, complete your assignment, get your certificate, and you can do your statistics on your own if you want to.
Okay, thank you, sir.
Yeah.
Does anybody else have any other doubts? You can raise your hand.
Yes.
Yes,
May I
Vijay.
ask one question, please?
Yes.
This is Vijay.
Yeah, Vijay, right.
Yes, sir. Please tell me how to authenticate the results or matter data given by AI.
Many journals
Hello?
recently, yeah,
That's
I've heard you.
that's
How to authenticate the data given by AI?
right, sir.
Yeah. Which data are you talking about? References or introduction or results or sample data, or which data you are talking about?
If we have given or feed AI with our data,
Okay.
which have got or results, any observations,
Okay.
and ask asked to analyze it.
Okay.
Uh,
You want that to, if, okay, you will give your data and you want the AI to give you the results part, give you the interpretation.
interpretation, results, observations.
Yeah. Okay. And you want to know how to authenticate the results that give by AI.
Definitely, definitely.
Yeah. So what you have to do is you have to interpret your results by yourself or with the statistician, and you have to compare it. It's like there is no other way to check with AI. You have to go give your results to your give the data and give your results to your certified statistician and compare the results with AI.
Exactly, exactly.
Yeah, that's it. There is no other way to authenticate. As far as I know, there is no other way to authenticate your data. Only a statistician can authenticate your data.
Yes, sir. Yes, sir.
Yeah.
Okay. Thank you so much. Thank you.
Does anybody else have any doubts?
One more question, sir.
Yeah, there's a go ahead.
Sir, may I know about case report? How to write like that?
Yeah, there's a your which batch, which year?
Third year, sir, starting.
Okay. So case reports. Case reports is like there is you can discuss about single case or you can discuss about group of cases.
Okay, sir.
Single case, what you have to do is like you will present, generally you will collect the data, you will present it in your wards, right, to your
Yes.
provinces. In the same way, you will collect all the required data, everything.
Yes,
There
sir.
are some guidelines to collect the case report data, to collect the data to publish a case report. It's like if you search Equator network in Google, Equator,
Okay. Okay.
yeah, you will find some guidelines how to report a case. So
Okay.
you will, it's like, so you have to collect all the data, the name, the demographic data you have to collect. The other way you will collect all the clinical data. What about the symptoms? All in a chronological order. You have to collect all the investigation details, everything. You have to collect A to Z. You should not miss anything. And
Okay,
then
sir.
you have to arrange it according to the case report format. Please go through that Equator network. You will see. You will get the format. In that
Okay.
way, you have to structure your case report. You have to follow the other guidelines. Every journal, many journals will have other guidelines, even for specific things. It's like they will have specific other guidelines for case reports, specific other guidelines from systematic reviews, that meta-analysis, specific other guidelines for original cases or whatever. So you have to follow the specific rules, and you can present your case.
For multiple cases.
It is
Same.
like, yeah, you can okay. Now I found some 10 similar cases. Now, okay, this is the condition, this is the similarity, this is the difference. These are the differences from previously found cases in somewhere else, and these are the differences that I have found in my case. You can discuss about all those these differences, similarities, all these factors that the variables that might have affected your case presentation, case outcomes, and you can present them.
Okay, sir.
Yeah.
Thank you, sir.
Yeah, thank you.
Anybody else has any doubts to ask, sir? You can ask any doubt, anything that you're struggling with in your current research, or any doubts that you have in today's session. So I think nobody has any further doubts.
Okay. Then, yeah. Thank you, guys.
Thanks a lot, sir.
Yeah. Thank you. Good night.